In this short video, I am going to discuss the cell morphology protocol, which is part of the cell biology protocol collection. To start off, select the count size ribbon and press the protocols button. In the cell biology collection, locate the cell morphology protocol and press the load button. All protocols come with demonstration images. To load the demonstration image, select the load demo next to the reset button with the open demo image checked. The protocol is based upon two dyes. One dye will be a nuclear dye to identify the nucleus, and a second dye to identify the cell morphology. This could be a cytoplasmic dye or, for example, a whole cell dye. Next, select the interactive checkbox, press the next step button, and we'll select the nucleus. In this case, we're using smart segmentation, which is a machine learning segmentation method. It's already been set up for the demonstration image. However, for your own images, you can use this protocol, or you may need to include more reference objects or backgrounds. Once the nuclei have been identified, press the OK button. There are some options to set a minimum size area. So if you have any cell debris or nonspecific fluorescence, you can filter that out. If you want to not count cells that are touching borders, you can select any of the options here for removing anything touching the borders. In this case, you normally select all. Fill holes. These nuclei are very nice and solid, so we don't have any gaps, so you don't need to fill those in. And they're well separated in this case, so we don't have to run any type of object splitting. I'm going to press the apply button, press the next step button to continue. And now we're going to segment the fluorescence identifying the cell outlines. In this case, it's the Psi5 channel. So you can see there's a little fluorescence here. And now I'm going to go in, because we're using smart segmentation machine learning, I'm going to train it a little further and start getting that cell shape. That's pretty good. You can see there's a little background fluorescence, but since there's no nuclei, when we go to merge everything together, this will be discounted automatically. That looks pretty good. I'll press the OK button. As you can see now, to identify a cell, it must contain a nuclei, and so we've done that. We do include, again, the options for minimum area, clean borders, fill holes, and object splitting. In this case, all cells are completely within field of view, so we do not need to apply clean borders to get whole intact cells. Press the next step button. And now a range of parameters have been calculated. I want to press the finish button for a second. And now, to give an example of the data that's collected with the cell morphology protocol, we'll calculate the area, aspect ratio, a range of morphometric parameters, a number of texture measurements, clumpiness, diameters, intensity information, again, more texture measurements, and a range of other parameters to describe the particular cells you've identified. And we also include statistical information besides the individual measurements. At this point, you could export the data, or as always, if you're running a multi-well plate, you could run a batch analysis, or if you have images residing in a folder, you can run a batch analysis on those too.